Hello everyone, this is chapter 6 on how enzymes work from Essential Biochemistry, and it covers sections 6.2 and 6.4. So I wanted to pick up from last time and talk about covalent catalysis. And in covalent catalysis, what happens is an enzyme has an amino acid side chain that acts as a nucleophile, and that reacts with an electrophile on the substrate. And what happens is the nucleophile donates 2 electrons, which can form a bond, and this forms a temporary covalent bond between the enzyme and the substrate. So imagine that you have a substrate, let's call this AX, and we can have a functional group that's X that's transferred to the enzyme. So now the enzyme is temporarily covalently bonded to that functional group X, and you release A as your first product. And then in the second step, the X is transferred to B, and then the release of the second product occurs, and then our enzyme is regenerated. So overall, what happens is that this functional group X is transferred to B, so now B has that functional group. So what that means is that this would just be the very simplest case, of course, but that we have 2 steps, and we go through 2 transition states, which are these peaks right here, and that the intermediate has an intermediary free energy. The energy is still pretty high compared to the reactants and the products, but it's not nearly as high as the transition state. So this would be the free energy of Xe right here, because the Xe is the intermediate. So let's look at some examples. So papain is a member of what we call the cysteine protease family. So the active site contains histidine and cysteine, and there are other members of that family. So once you learn the mechanism for papain, that means you know how all cysteine proteases work. And this is a protease, and proteases catalyze the hydrolysis of peptide bonds in proteins. So that means you have a protein as one of your substrates and water as another substrate. And the hydrolysis will occur at the peptide bond, and you will generate your carboxylate group and an amino group. And this is where the, in red, you can see where the H2O is donated to form your 2 products. So the way that this works is that if you go over here on the left, this is supposed to represent the enzyme right here. So the nucleophile in the case of papain is a cysteine. So it just shows X right here, sort of as a generic mechanism, but that's going to be S, sulfur, with its lone pair of electrons. So it's going to be unproteinated. It's going to be cis minus. And it attacks the carbonyl in the peptide bond of the substrate protein, and then these electrons will swing out over here to the oxygen, and now we have a tetrahedral intermediate. So the substrate protein is temporarily covalently bonded to the enzyme. The next thing that will happen is if we have an acid catalyst, here we have a histidine, so the histidine is going to donate a proton to the cis-acyl bond. So this right here, that's the peptide bond, the cis-acyl bond. The cis-acyl bond is the bond that we're breaking. So the electrons in that bond form a bond with hydrogen, and that effectively breaks that bond. And then the electrons swing over here, and now we release our first amine product. So we're hydrolyzing the protein into 2 pieces, and we release the first piece. There's another piece still covalently bonded to the enzyme. So this forms sort of a thioester or an acyl enzyme adduct. So again, we're still bonded to the enzyme. So now we're going to bring in our second substrate, the water. The histidine is able to grab the proton of the water, making this an OH minus, a much more effective nucleophile than water. And we are going to nucleophilically attack this carbonyl. Those electrons will swing out over here. And now we have our second tetrahedral intermediate over here on the right. So the next thing that happens is that the electrons then go back to the sulfur, and that breaks the bond between the substrate and the enzyme. And now we reform our carboxylate group, and then we have our product. And we regenerate the enzyme. Remember, the enzyme was such that the sulfur on the sulfhydryl group was unprotonated on the cysteine, and the histidine was protonated. So what that means is that if we look at papain and we carry out a reaction at various pHs, we're going to find that the pH optimum of this reaction is 6.2. And there's a reason for that. If we look at these inflection points, this tells us the state of protonation of the cysteine and the histidine. the pKa or the pKr of the cysteine is very low. In the protein, it's only 4.2, but if we're at a pH optimum of 6.2, the pH is higher than the pK, and that means the cysteine is unprotonated. So it's a CH2S minus. Whereas if we are looking at the histidine, its pKa value is 8.2. And if we're at a pH of 6.2, the pH optimum, that's a pH lower than the 8.2, and the histidine over here. Oops, it looks like I drew that as protonated. The histidine should be it's protonated. Yeah, no, I drew that right. Okay, so the histidine over here is protonated. We can figure out the pKa values of these groups by looking at the inflection point. So the inflection point here on this curve is 4.2. Here this pH 8.2. So the pKr of the cysteine is 4.2. The pKr of the histidine is 8.2. At a pH optimum of 6.2, the cysteine is unprotonated and the histidine is protonated. And if you go back and look at the mechanism, that's the state it has to be in order for the mechanism to occur. And that's shown right here. And I'm not sure why the pKrs are shown in different values, especially since I got this from the same book. But in any case, right here in the middle, we're at the sweet spot where we have an unprotonated cysteine and a protonated histidine. Because if we were at a really low pH and both of these residues were protonated, then the cysteine couldn't act as a good nucleophile with the proton there. Whereas if we were at a high pH and both residues were unprotonated, you could get started because the cysteine would act as a nucleophile, but then that step where the proton from the histidine is donated to the cis-al bond, that wouldn't happen. So you would not be able to complete the reaction. So in between is what we need: an unprotonated cysteine and a protonated histidine. So if we're looking at enzymes in general, these are the kinds of residues that could serve as nucleophiles: serine, tyrosine, cysteine, lysine, and histidine, and there are others, but these are a lot of the common ones. But we should note that, not in this form, that they have to lose their protons right here to get a lone pair of electrons. So this over here on the right, that's the state at which they have to be a nucleophile. So that means there has to be some other amino acid side chain in the enzyme that grabs a proton that converts this into this nucleophilic form. And again, it doesn't have to be negatively charged. A lot of them are, but you do have to have a lone pair of electrons because those are the electrons that are donated to form the bond, that temporary covalent bond between the enzyme and the substrate. So we can look back at this table as we are learning our enzyme mechanisms, so we can see the kinds of amino acid side chains and the kinds of things they might do based on their charge or their ability to act as nucleophiles or to transfer protons or to bind anions. So we should come back to this table and kind of fill in our knowledge there. The kinds of electrophiles you see in biochemistry are protons, metal ions, this carbonyl group, of course, and this is something called a Schiff base, and this carbon right here that's shown in red is susceptible to nucleophilic attack. So let's look at an example of metal ion catalysis. So there are lots of metalloenzymes, and they have metal ions, and they play an important catalytic role because they do things that lots of enzymes do, is that the metal ions help to position the substrate properly. You need to orient them so their functional groups are nearby so they can react to form the product. But they can also mediate redox reactions because metals can have different oxidation states, like iron can be Fe2+ or Fe3+. And because they're positive, they could stabilize or shield negative charges on the transition state. And remember, all enzymes stabilize the transition state. That's how you speed things up, by lowering the activation energy of the transition state. That's how you speed up a reaction. So carbonic anhydrase is an enzyme we're familiar with. We know that we take water and CO2 to make carbonic acid. And this is what the active site looks like. It has this cool, these beta sheets right here. This sort of cyan blue kind of color right here, that's the zinc. And it's covalent attached to 3 histidines. And here, they're not using CPK coloring, but the carbon is green, and then there are 3 oxygens. So this is a bicarbonate right here bonded to the active site. So the way that this happens is that water is a substrate, so zinc is going to polarize the water to form an OH-, and then that can nucleophilically attack the CO2, which is the second substrate. So let's see how that happens. If we look over here on the left, we can see zinc, and it's covalently attached to these 3 histidines. There's another histidine over here, sort of on the far left, that's not bound to the zinc. You can see these are 3 red spheres. Those are all supposed to be water molecules, so they're just showing the oxygen and they're not showing the hydrogen. So it's supposed to be H2O, but they're just showing the O. But what happens is the histidine 64 grabs a proton from this water, which grabs a proton from that water, which grabs a proton from that water. So you end up with, if you look over here at the top, zinc bonded to an OH minus. And this IM stands for imidazole, and that's the name of those 3 histidine rings. So first step, the OH minus acts as a nucleophile, nucleophilically attacks the carbon on the CO2. And then, so that's the CO2 is our first substrate. And then we're going to bring in our second substrate, the water, and that comes in and replenishes the OH minus because we need to regenerate our reaction. And we also donate a proton, and then this is our bicarbonate. So I show this as H2CO3, which of course dissociates into H plus and bicarbonate. So that's how zinc is involved in that reaction. So let's move on to the serine proteases. They're similar to the cysteine proteases. They're called serine proteases because they all have an essential serine. I'm going to show you how chymotrypsin works, and that means that once you know how that works, you know how all the serine proteases work. So, but first I want to do some experiments where we explain how do you know what the amino acids are on the active site. This is before you have crystallized the enzyme. Well, one of the things you can do is you could add chemical reagents, and they will react with active site amino acid side chains when other reagents won't. So, for example, if we look at chymotrypsin, and we have a serine, so here's the serine. This is serine number 195, which is in the active site. We can add DIPF, diisopropyl phosphofluoridate, and that forms that carries out a reaction where we form a covalent bond. And now the serine has this big group attached to it, and that means the enzyme's inactive. What's interesting about this is there's lots of serines in chymotrypsin, but that's the one that's the most reactive. So that tells me, oh, serine 195 must be in the active site. We can do another reaction where we take something called tosylphenylalanine chloromethyl ketone, and I'm just drawing it like this in the red, and that's the R group right there. And the TPCK will again put a covalent group onto the nitrogen of the histidine, and that also inactivates the enzyme. And again, there are lots of other histidines in chymotrypsin, but the histidine 57 is the most reactive. Okay, so here's our catalytic triad. We have aspartate, histidine, and serine. So asp102, his57, serine 195. They're maybe far apart in linear structure, but when they fold, they form the active site. So I have a video on Canvas. If I click on this link right here, I'm not going to do this right now because in my screen capture, the videos tend not to turn out very well. So I'm going to direct you to that video. So instead, I'm going to just walk you through the still pictures in our book. So here's the aspartate, the histidine, and the serine. And what happens is the aspartate kind of polarizes the histidine, which grabs a proton from the serine. So now instead of CH2OH or CH2O minus, and then that can nucleophilically attack the carbonyl of the peptide bond. So this is our polypeptide substrate right here in blue. And now what we have is here's our serine, and we have a temporarily covalent bond between the serine and our substrate, which is shown in blue. So the next thing that happens is these electrons will swing in, and then this is the scissile bond. That's the peptide bond we're going to break. And the way we can break that is those electrons will form a bond with this hydrogen. So the histidine is the one that gives us that hydrogen. And when we do that, now we're down here, and this bond right here, the scissile bond, is now broken. And now this is our first product. So we're cleaving this protein into 2 pieces. That's 1 piece. And then that can leave. But still, here's the serine. And now we have an ester or an acyl enzyme intermediate right here. Okay, so what happens next? Well, what happens next is that we do release this product. So that product is gone. And now on the serine, we have our acyl intermediate. We're going to bring in our next substrate, which is water, shown in green, and the histidine is going to grab this proton from the water, making this an OH minus better nucleophile. I'm going to attack this carbonyl, and what happens when I do that is I form a bond. So now I've got a tetrahedral intermediate again. And to finish this off, I'm going to break the bond between my substrate and my enzyme. So I need these electrons right here. I'm going to form a bond with this hydrogen that I'm going to grab from the histidine, and then that's going to release my product. And there's my product right there. And I have regenerated my enzyme like all good enzymes are. So one of the things that we have to do when we're thinking about this mechanism is that, remember, this is what all enzymes do. All enzymes stabilize the transition state. All enzymes do that. And one of the things we can do is we can look at what happens when we form these tetrahedral intermediates. So this step shows the formation of the tetrahedral intermediate. And notice that the tetrahedral intermediate had an O- charge right here. Well, the serine 195, and I don't know what, there's a 194 and 193, I don't remember what amino acid that is, but we have NHs that are part of the peptide bonds there, and they can form hydrogen bonds, which are shown by little green dots. And that will stabilize this tetrahedral intermediate, and the tetrahedral intermediate resembles the transition state. So here are these, another role of the enzyme is to stabilize the transition state. So these NH groups as part of the peptide bond and the oxyanion hole will stabilize the transition state. So that now we know how chymotrypsin works. That means trypsin and elastase work the same way. In fact, we can superimpose trypsin and elastase on top of each other. So chymotrypsin is blue, elastase is red, trypsin is green, and the catalytic mechanism is all the same, but their substrate specificities are different. So there must be something different about them. We remember from chapter 4 that chymotrypsin cleaves on the carboxylic side of the trypentire, the aromatic amino acids. For trypsin, it's the positively charged basic amino acids lysine arg, and for elastase, it's guanyl and maybe valencyr, so small amino acid side chains. So if we look at, this is trypsin, so if we look at trypsin in the middle, we can see that we just have the backbone trace right here, and in stick form, we have the aspartate 102, the histidine 57, and the serine 195. And in cyan, that's our substrate. And remember, trypsin cleaves on the side of lysine and arginine. And what's being shown right here, I can't exactly see. I think it's probably lysine. Maybe it's an arginine, it's hard to tell. But it's one of those two, and it's fitting into a nice pocket that will fit a basic amino acid really well. So it turns out that what's different about these serine proteases is the pocket. So for chymotrypsin, there's a pocket that fits a flat aromatic ring really well. For the trypsin, there's an aspartate at the bottom, so a positively charged side chain will form a nice ion pair there. For the elastase, you have a valine and a threone that kind of stick out and make this pocket a little narrow, a little shallow, so only small amino acids can fit. So this explains why we have the asp-his-ser, that everybody has that same catalytic triad. There's the active site, here's the oxyanion hole, but this pocket is different. So this is chymotrypsin, so chymotrypsin's pocket's going to be hydrophobic, but trypsin's going to have the aspartate, and the elastase has a shallower pocket. So I wanted to come back to why these enzymes exist in the first place and why Mother Nature made them. She didn't make them for us. The purpose of these enzymes is to digest the proteins in our food, and this is not to draw to scale. But this big yellow blob right here is the pancreas, and this blue thing right here is the pancreatic duct. So when we eat a meal, we chew our food and it gets to the stomach, and protein digestion starts in the stomach a little bit, but most of it happens in the small intestine. So we're going to need enzymes to hydrolyze the peptide bonds in the big proteins that we eat down to amino acids so that they can be absorbed by the cells lining the small intestine. But since these enzymes digest proteins, we don't want to be digesting away our pancreas. So the way that this works is that when the enzymes are traveling down the pancreatic duct, they have to be inactive, and you can't activate them until they get in the small intestine, because that's where they're needed, and that way you won't damage anything along the way. So there's a cascade where we take trypsinogen. So trypsinogen is the inactive name for this. Enteropeptidase is an enzyme that's waiting for it in the small intestine. We say like entero, that means small intestine. So the enteropeptidase activates the trypsinogen to trypsin, and then trypsin can go and activate more of its neighbors. It can also activate chymotrypsinogen, which is the inactive form to chymotrypsin, and the proelastase, that's the inactive name for elastase, and then it can activate it. So this has a cascade effect where once you activate one trypsin, you can activate tens or dozens or hundreds of chymotrypsin and elastase. So how does this work? This is an old scan from one of my old biochemistry books. So what we're going to do is we're going to just do a cleavage of a couple of sites. And somehow when the protein folds and forms this inactive precursor, this zymogen is inactive. If you snip out a little piece right here, you can you can have the active site fall into place. So how does that work? Well, this is a very stylized form right here, but this shows chymotrypsinogen. It's a single chain from 1 to 245 amino acids. And what we're going to do is this series of trypsin and then chymotrypsin that's activated, we're going to hydrolyze some peptide bonds. And essentially what we end up with is we get rid of this piece 14 and 15 and 147 and 148. So now we have 3 pieces. Do not worry, they will stay together because they're disulfide bonded. That's not shown here. But the fully intact 245 amino acid protein is inactive, so it's not until you take these pieces out that it becomes active. Well, how does that happen? Well, I'm going to look at trypsinogen as an example. So for enteropeptidase, we're going to cleave between lysine and isoleucine right here, between 15 and 16. And that's what the peptide bond looks like. So this would have been the lysine over here. This is the isoleucine over here. When you do a hydrolysis, what happens is you now get, instead of a neutral peptide bond, if you add water here, you're going to get a carboxylate group here and an amino group here. So now you have charged species where you didn't have them before. So let's zoom in on this isoleucine right here. That has an NH3+. Remember over here when the peptide bond was intact, it's not positively charged. So what happens is, look down here at the bottom on the left. Here's isoleucine where it's blue. That's the NH3+. And the isoleucine forms an ion pair with asp-194, which just happens to be next to Ser-195. And then that kind of pulls the his and the 102 into place, and now we have our active triad. So how does that work? I have 2 views right here that I'm going to attempt to animate. At the top, up here on the left, is the chymotrypsinogen, the inactive form, and this yellow stick right here, that's the isoleucine that's part of the peptide bond. But then when you cleave that peptide bond, this can swing out over here, and there's that asp-194 right there. So we're going to form that ion pair. So I'm going to give you these again. So here is chymotrypsinogen in red. This is the serine, that's the histidine, and that's the aspartate, and that's the catalytic triad, and they're not quite lined up, so the enzyme's not active. There's asp-194 right there, and there is the isoleucine number 16. So this whole thing is intact. We haven't done a hydrolysis. But when we do a hydrolysis, watch what happens. When we do a hydrolysis and we break this peptide bond, so now we have a break. Now there's the isoleucine with its NH3+, and now it's forming an ion pair with the asp-194, which is next to the Ser-195, and now this is lined up. So if I animate this for you again, so inactive, active, so inactive, active. So again, look at what's happening here, where just that cleavage produces that NH3+, and I'm going to go way up here to form the ion pair, and then that lines that up into place. So Mother Nature also looks out for us in various ways. So now suppose you had something wrong in your pancreas, and these inactive zymogens started to activate. We have something in our pancreas called pancreatic trypsin inhibitor. So this is trypsin, and there's something that the pancreatic trypsin inhibitor looks like a substrate, but it has a Ki that's really, really low. And what happens is the pancreatic trypsin inhibitor binds to the enzyme, the prematurely activated enzyme, and forms an enzyme substrate complex that's so stable that it never forms product, and it plugs up the active site so other proteins can't bind. So that's sort of insurance policy so that we don't destroy our pancreas if some of our enzymes get prematurely activated. So that was our whirlwind tour of enzyme mechanisms. Please watch the videos on Canvas, especially the chymotrypsin video. It goes into a lot of detail. I think you'll find it really helpful. Thank you for your attention, and I will see you in class.